Hello, everyone. I'm Chau, and I'm currently a student at Aalto University in Finland. And I'm going to present our work on incorporating hierarchical information into multiple instance learning for modeling patient phenotypes with single-cell RNA sequencing data. So single-cell RNA sequencing data has the potential to improve the prediction of patient-level characteristics, such as disease phenotypes and clinical outcomes, by providing high resolution and disease-specific signatures. And in recent years, several methods have been developed for the prediction of those patient-level characteristics using single-cell data. And among those, multiple instance learning, or MIL, emerges as a promising framework due to its ability to structurally model the cellular heterogeneity. However, existing MIL models do not account for the hierarchical structure of single-cell data, especially the biological groupings of cells or cell types, which can lead to suboptimal performance and limited interpretability at the cell type level. So to bridge this gap, we introduce an approach to incorporate hierarchical information into the attention-based MIL framework. We'll get started with some background information on MIL and relevant MIL models. So MIL refers to a specific setting of supervised learning where data is organized into bags, and each bag contains several instances. The key premise of MIL is that only the bag level labels are known and the instance level labels are unknown. Therefore, the task of MIL models is to predict labels of bags using features of their instances. And in our setting, the bags are the biological samples from patients or donors, and the instances are the cells. So one key consideration in MIL is how to aggregate the instances into the bag representation for prediction. Else et al. Introduced the attention-based aggregation technique, which models the bag representation, Z here, as an attention-weighted sum of the instance representations, HK, and the attention weights, AK, are modeled as a transformation of the HK followed by a softmax activation over the instances. So the attention weights can be interpreted as the relative importance of the instances. So this aggregation technique allows MIL models to capture the cellular heterogeneity of single-cell data and also provides interpretability at the single-cell level. And building upon this work, Engelmann et al. Proposed mixed MIL, which is a MIL model that integrates the attention-based aggregation technique with a generalized linear mixed model, or GLMM. And to be more specific, mixed MIL aggregates the cell representations X here into the sample representation Z gamma with the attention technique that models the dependency between Z and the label mu with a GLMM. And the attention function omega is a shallow function consisting of a linear transformation of X and a softmax over the cells. So we present an approach to incorporate hierarchical information into the aggregation step of the MIL framework. Our approach performs a step-by-step aggregation process, first over cells to produce cell type representations, and then over cell types to produce the sample representations for prediction. And we show that the final prediction can be decomposed into a sum of either cell or cell type contributions, so providing interpretability at both the cell and the cell type levels. And based on this approach, we present two models. the cell type attention or CTA model with attention only on the cell types, and the hierarchical attention or HA model with attention on both cells and cell types. So this figure shows an overview of our models, starting with the cell representations annotated by their cell types. We aggregate the cells to produce the cell type representations, and then aggregate the cell types to produce the sample representations for prediction. We'll now take a closer look at the model for a single sample with label Y. Our model inputs single cell data D, which consists of either the gene expression or possibly lower dimensional representations of the cells in the sample. And the architecture of our model is quite simple. It has a few feed-forward layers in the beginning to reduce the dimensions of the input. Next is the two-step aggregation process to aggregate cells to cell types and cell types to sample for prediction. And the sample representations are then passed through a linear classification layer to obtain the prediction. And note that the first aggregation step from cell to cell types is attention-based aggregation for the HA model and mean pooling for the CTA model. So this step is the only difference between the two models. This is the formulation of the first aggregation step. So in this step, the HA model performs an attention-based aggregation over the cells, starting with the representation Hij of cell J within cell type I. We compute a cell-level attention weight, alpha Ij, using a linear transformation of Hij followed by a softmax function within the cell type. And for each cell type I, we then compute a cell type representation Hi by aggregating Hij using the attention weights alpha Ij. Alternatively, in the CTA model, the cell type representations Hi is obtained via mean pooling of the cell representations Hij, and the factor alpha Ij is now defined as 1 over the number of cells in cell type I. In the second aggregation step, we compute a cell type attention weight beta I using a linear transformation of cell type representations Hi, followed by a softmax function over all cell types in the sample. And for each sample, we define a sample representation H by aggregating Hi using the cell type attention weights beta I. And this last transformation shows that H can also be written as a weighted sum of all the cell representations Hij in the sample with weights gamma Ij defined as the product of alpha Ij and beta I. Finally, during prediction, the sample representations H are mapped to the labels using a linear function followed by a softmax over the label classes. We now show that the logits of the final prediction can be decomposed into a sum of either cell or cell type contributions, making the model interpretable at both levels. So if we let W times H equals Z, then the prediction logits before softmax are just Z shifted by some bias B. Then Z can be decomposed into a sum of cell logits or cell type logits, and these logits represent the contribution of each cell or cell types toward the final prediction. And since we know the cell type contributions, we can identify the critical cell types that are most relevant for prediction. and these cell types likely play a role in the mechanism of the disease phenotypes or clinical outcomes. So to identify these critical cell types, we first decompose the prediction logics of each sample into a sum of cell type level logics using the formulation shown in the previous slide. And here the logit of cell type I in sample S is denoted as LSI. And in the case of binary classification, a positive cell type logit indicates contribution to the positive class, while a negative logit represents contribution to the negative class. Therefore, on average, cell types with higher logits in positive samples and lower logits in the negative samples contribute more to the correct prediction. And based on this intuition, we introduce a metric called importance score, kappa I, to measure the overall contribution of cell type I in differentiating between the phenotypes. And we use a permutation test to identify cell types with a statistically significant importance score. Specifically, we repeatedly permute the patient labels, train the model on this permuted data, and extract the importance scores for each cell type. This results in a null distribution of importance scores for each cell type, from which we can calculate the empirical p-values and identify the critical cell types. We evaluate the performance of our approach on 3 datasets, cardio, COVID, and ICB. On the cardio dataset, the task is to classify between healthy controls, patients with hypertrophic cardiomyopathy, and patients with dilated cardiomyopathy. On the COVID dataset, the task is to classify between healthy controls and patients with COVID. And on the ICB dataset, the task is to predict whether cancer patients respond favorably or unfavorably to the immune checkpoint blockade treatment. We consider 5 baselines where, except for the general purpose random forest classifier, the other 4 models are also methods developed for patient phenotype prediction with scRNA-seq data. So we first evaluate the prediction performance of all models on the 3 benchmark datasets using a repeated nested cross-validation procedure. And this table here presents the AUC values of the models, and the 2 highest AUC values for each dataset are in bold. These results demonstrate the competitive performance of both of our models. Next, we investigate the sensitivity of our model to several factors in data quality, including variations in training sizes, cell counts, and cell type annotations. So first, to test the robustness of models to varying training sizes, we evaluate models on 100 train test splits with 25, 50, and 75% of samples used for training. To investigate the effects of varying cell counts on model performance, we perform nested CV, but randomly subsampling cells in each sample such that 25, 50, and 75% of cells in each sample are included. And finally, since our model inputs cell type annotations, we evaluate the effects of noisy annotations on model performance by conducting nested CV with 25 and 50% of cells in each sample being assigned random annotations. These are the results of the 3 experiments. We observed that for all models, the effects of low cell counts seem less pronounced than that of low sample sizes. And moreover, the performance of our model declines as the cell type annotations get noisier, and the CTA model seems less sensitive than the AHA model. possibly because the mean pooling layer of the CTA model is more robust. This dependency on cell type annotation is a downside of our model, and hopefully can be alleviated with data augmentation techniques. And next, to validate the benefits of incorporating the cell type information into the MIL framework, we conduct an ablation test. Specifically, we compare the performance of our two proposed models with two modified models that do not input the cell type information. In the mean pooling model, the entire attention-based aggregation module is omitted, and the sample level representation, H bar here, is generated by mean pooling the cell level representations, Hij. And in the cell attention model, only the cell type attention layer is omitted, and the sample level representation, H tilde here, is an attention-weighted sum of the cell level representations, Hij. The nested CV results are given in this table. So our model achieved the highest mean AUC on all except the COVID dataset, where the mean pooling model also achieves the highest AUC. These results demonstrate the utility of incorporating cell type information into the attention-based MIL framework for modeling single-cell data. And finally, to evaluate the biological relevance of our model, we use the HA model to identify the critical cell types on the COVID dataset. The model detects 11 critical cell types consisting of mostly immune and epithelial cell types. And among these, ciliated secretory and goblet cells obtain the lowest adjusted p-values, and macrophages, ciliated cells, and secretory cells obtain the highest importance scores. So this emphasis on the importance of ciliated cells and secretory cells generally aligns with existing biological knowledge, showing that our model is potentially capable of capturing biologically relevant information. This is the end of my presentation. Please let me know if you have any questions. Sorry.
You hear me?
Yes. Yes.
Yes, I said great talk, and that my understanding of your method is that you're narrowing down the patient phenotype to the contribution of different cell types. But I can imagine that in different conditions, there might be other important factors that are coming from single-cell RNA-seq. So do you have a way to evaluate these residuals? So how much of if there is other effect that present in single cell RNA-seq data that is not relevant to cell types proportions or cell types influences?
Yeah, thank you for the question. Yeah, there are possibly other like factors as well. For example, patient level like information like sex or like clinical history, and these like patient level information can like be incorporated into the model. like for example, after aggregating the cell representations into the sample representation. Yeah, but for our model, we are just mostly focusing on modeling the hierarchical structure of single cell data.
Yeah, I was just saying that there might be other effects in single cell data other than cell types that might be also relevant for the prediction task.
Yeah. So can you give an example? Because
maybe you can discuss later.
Right.
Yeah, thank you. So